Hello everybody, welcome back to another exciting edition of Ed Puzzle Lecture Notes. Today we're going to go over the anatomy and physiology of the pancreas, located right there. The stomach has been removed, and also the liver. Now these are considered accessory structures because food does not directly pass through these structures, but they do aid in digestion, just like your tongue, your saliva, your teeth, etc. Now, what you need to know that the pancreas is considered both endocrine and exocrine in nature. In its endocrine function, it's releasing its contents, its hormones, directly into the bloodstream. Now, in its exocrine function, it's releasing its contents into the gastrointestinal tract or the alimentary canal. Now, even though that's still in your body, that canal is eventually going to leave your body, so it's considered exocrine. Now, when the pancreas is directly involved in digestion, its contents go through what's called the pancreatic duct. And you can see that right here. And you can see it intersects with what's called the common bile duct, which is coming from the liver. And both are going to excrete their product into the duodenum, the very first section of the small intestine. Now also notice, even though you're not going to know that it's 4 inches below, but also know that it occurs what's below what's called the pyloric sphincter. And that is a valve between the stomach and the very beginning of the small intestine called the duodenum. And now the histology of the pancreas, or the tissues and cells that make up the pancreas. First is called the acini, or dark clusters, and that is all of this stuff out in here. And that is the part of the pancreas which is exocrine. You will not need to know that it makes up 99%. You should know that it makes up by far the majority of the pancreas. So this is the part of the pancreas which is exocrine, which is releasing its contents, its pancreatic juice, into the duodenum of the small intestine that helps in digestion. Now the endocrine portion of the pancreas, located right here, is the lighter part called the islets of Langerhans. That is what's involved in blood glucose regulation. So you have what are called your alpha cells. When blood glucose gets too low, they release what's called glucagon. And that's going to cause the liver to convert glycogen into glucose, which will increase the level of glucose in the blood. Now the opposite of that is, if blood glucose gets too high, there's other cells called beta cells, which then secrete insulin, which is going to bring glucose levels down. It's going to cause the muscles to start storing glucose as glycogen, which will decrease the amount of glucose in the blood. Now this can be hard to remember, but the way I remember it is alpha cells, A comes before B alphabetically, and also G comes before I. So alpha cells secrete glucagon, beta cells secrete insulin. And the pancreatic juice. You're not going to need to know how many quarts a day it's going to be, but you should know that it's considered basic. I don't know if you remember from regular biology, but anything below 7, so below 7 is considered acidic. Everything above pH-wise, higher than 7 is basic. Now the stomach is very acidic, so this pancreatic juice has to be basic, or the contents of the stomach is going to burn right through the small intestines because the small intestines is not ready for acidic fluids that's coming from the stomach. So this pancreatic juice contains sodium bicarbonate, which is kind of like baking soda. It's a base which decreases the acidity of the contents coming out of the stomach, which protects your small intestine. Now, you're not going to know that it's clear. You're not going to know that, or it's colorless. You will need to know that it does contain enzymes. That's extremely important. And here are some of the enzymes it secretes. For carbohydrates, now in the mouth we had salivary amylase, which is in your saliva, but in the small intestines, the pancreas secretes pancreatic amylase, which breaks down carbohydrates into sugars. It also has pancreatic lipase, which lipids are fats, so that breaks down fats. It also secretes 3 enzymes, which helps to break down proteins: trypsin, chymotrypsin, and carboxypeptidase. Know all 3 of those. I guarantee you will see that on your exam. Now it also secretes enzymes which break down nucleic acids, ribonuclease and deoxyribonuclease, but that will not be as important as the enzymes it secretes for proteins: trypsin, chymotrypsin, and carboxypeptidase. Definitely remember those. Don't worry so much about the enzymes it secretes to break down nucleic acids. And now this is how the pancreas and the liver work on blood sugar levels, keeping it in a balance, homeostasis. Now remember, negative feedback. That means if homeostasis, if it starts to get too high, negative feedback means it's going to bring it back down into homeostasis. And the opposite of that, if it starts going too low, homeostasis, negative feedback is going to bring it back into more of a balance. So this is a lot of words here, and there's going to be a picture on the next slide. But if blood glucose levels get low, that's called hypoglycemia. Now when that happens, the pancreas secretes glucagon. Now remember, that's from the alpha cells, because the other substance, the other hormone that the pancreas secretes is called insulin. Remember, G comes before I alphabetically, and so does A comes before B. So alpha cells secrete glucagon. Now the liver is the main storage of glycogen. That's where animals store their glucose in a carbohydrate called glycogen. The liver is the main storage of this. Glucagon tells the liver that the blood glucose is too low, and it tells the liver to start converting to break down glycogen into glucose and release it into the blood. And obvious when this happens, when liver releases glucose in the blood glucose levels will increase. Now the blood glucose gets too high, which is called hyperglycemia. That's going to inhibit the release of glucagon from the pancreas. And if blood glucose gets too high, it's going to tell the pancreas, the beta cells, to secrete insulin. Now the hormone insulin tells your skeletal muscles and your liver to convert glucose in the blood and to turn it back into glycogen so it can be stored. And that'll bring the blood sugar back down. And here's a little diagram that shows you the process to keep blood sugar level in homeostasis. And when it's in homeostasis, you have about 90 milligrams of glucose per 100 milliliters of blood. So now, if the blood glucose starts to get too low, your pancreas, if glucose is low, alpha cells, remember A comes before B, will secrete glucagon. Now that tells the liver, which is the main storage of glycogen. Glycogen is how animals store glucose. They store it as glycogen. They also store it as fat. That's the main energy storage. And it tells it to turn that glycogen into glucose and release it in the blood. And that'll cause the blood glucose levels to rise to make it back into homeostasis. Now the opposite of that is if blood glucose levels start to get too high. That is where the beta cells will notice that, and they'll secrete insulin. Remember, B comes after A alphabetically, and I comes after G alphabetically. That's where insulin is secreted, and that's going to tell your muscles to start taking in glucose and storing it as glycogen, or using it, burning it, and also tells your liver, take the glucose from the blood and store it as glycogen. So since they're storing it as glycogen, that's going to bring the blood glucose levels down and return it back to homeostasis. All right, briefly, the histology or the tissues of the liver. You will not need to know all of this, so I'm just going to emphasize what I think you're going to need to know. You definitely need to know that liver cells are called hepatocytes. Those are the functional unit, the functional cells of the liver. You will definitely need to know that the liver secretes bile, and it has bile canaliculi, which are basically just like small little channels, which are going to meet up to form a larger duct, which the bile is then secreted into the duodenum of the small intestines. So you'll need to know it does produce bile, but I doubt that you're going to have to know precisely like canaliculi. Hepatic sinusoids, just the blood capillaries between the hepatocytes, not really important. Stellate reticuloendothelial cells, I always call them just Kupffer cells. These are phagocytes, so these are the ones that they're kind of like little cells that eat other things, so they destroy worn out blood cells, microbes, and foreign matter. So those are kind of like the immune system of the liver. And now the little helper of the liver called the gallbladder. You always see that. It's always right there underneath the liver, and it's always green. You're not going to need to know what cells, but it's simple columnar epithelium, which means it's a single layer of cells. They're not stacked on top of each other, and they're not like that, but they're kind of like columnar in shape. 3 layers of smooth muscle. You don't need to know that. And the serosa or visceral peritoneum is wrapped around them. So here's your gallbladder right here. And this is the common bile duct. That's going to go to the small intestines, the duodenum. Here's all those canaliculi in the liver. They come together to form the common hepatic duct. And then that together, that goes to the duodenum, the first part of the small intestines. But the main thing you need to know, the gallbladder is what stores and increases the potency of bile, and then it secretes it into the small intestine. All right, now here is that substance called bile. Yellow, greenish liquid. It's made of water, bile salts, cholesterol, lecithin, and pigments. Here's its function. This is what you're going to need to know. Emulsification. So it's secreted into the small intestine, the duodenum, the first part of the small intestine. And that's what makes large fat molecules. It breaks them down into the smaller triglycerides so they can be absorbed in the small intestine. If it wasn't for bile, those large globules of fat, of lipids, would be too large and they would pass right through the digestive system without being broken down and absorbed into the blood in the small intestines. And a little silly looking word right here, bilirubin. I guarantee that'll be on the test. It's not going to be like a short answer question, but it'll probably be one multiple choice question. And this is the pigment which is produced in the liver when the heme in red blood cells is broken down. So basically, it's the part of the red blood cells that carries the oxygen. Well, when they get worn down, they need to be broken down, they need to be metabolized and gotten rid of, and that is the liver that does that, and it produces that pigment called bilirubin. And it's that bilirubin that gives feces its brownish color. It also gives urine its yellowish color, depends on where it's being excreted. Is it being excreted through urine, which is filtered out by the kidneys, or does it go straight through the digestive tract and given out as feces? But that's bilirubin. It's the pigment that is produced when old red blood cells are broken down. And now some functions of the liver. Now this could turn into a short answer question. Now, I guarantee you're not going to have to know all of these, but definitely become a master of like 3 or 4 of them. Number 1, super important, and I think it's one of the easier ones to remember. Metabolize carbohydrates to help maintain blood glucose levels. So we remember if there's excess glucose in the blood, the liver is going to turn that and store it as glycogen. And that is the long-term energy storage in animals. Plants store it as starch or oils. Animals store it as glycogen or fat. And some of the glycogen is stored in muscles, but a large part of it is stored in the liver. So when glucose levels are high, the liver stores the glucose, converts it into glycogen, and stores it. Now when glucose gets too low, that glycogen can be broken down by the liver and converted back into glucose and released into the blood, which will obviously raise blood glucose levels. Another function is it helps to metabolize lipids. I'm not going to go over all these 4 bullet points, but just remember bile. It produces bile, which is stored in the gallbladder, and is that bile which helps break down fatty acids in the small intestines, but that bile is produced in the liver, so it helps to break down or metabolize lipids or fats. Now this is going to be a less important function of liver, not like, some are more important than the others, but this is one that's, it's not going to be on our test, I don't think. It's called deamination, which is a process which helps to break down proteins, and it does that by removing what's called the amine group, amino acids, or proteins are made of amino acids, and the liver removes this NH2, which allows the protein to kind of unravel and spread out, which then it can be broken down and metabolized so it can be used for energy. Now in the process of deamination, removing that amine group, it creates a lot of toxic ammonia. And then the liver quickly turns that ammonia into what's called urea, which then gets excreted by the kidneys and released in urine. And the liver also plays a role in synthesizing special proteins, which are important for blood clotting. That's basically if you start bleeding, those proteins like platelets help to clot the blood so you don't continuously bleed, that the wound will eventually heal. Very important process right here, number 4. The liver definitely is the main organ that processes, gets rid of harmful drugs and hormones that are no longer needed. So it detoxifies the blood by basically breaking down the drugs and alcohol to remove them from the body. Also, hormones like your thyroid hormone and estrogen, you keep making more of this, but you got to get rid of it eventually, and it's the liver which breaks it down and allows it to be removed from the body. It creates bilirubin. Remember, that's the pigment that is produced when aged red blood cells are broken down, and it creates that pigment called bilirubin. The liver is also a storage for mainly fat-soluble vitamins like vitamin A, B12, D, E, and K. And since the liver helps to break down fats, help to digest fats, it's also a key way that these important vitamins are ingested or absorbed in the gastrointestinal tract. It also helps to activate vitamin D. Vitamin D is very important when you take in calcium, so the health of your bones is very important if you have to have vitamin D, because you can't absorb calcium without vitamin D. Also an hour of sunlight, and the liver doesn't really do this alone. It works with the pancreas to fully activate vitamin D so calcium can be absorbed, and calcium is important to maintaining proper bone health. And that is it. Those are the functions of the liver. All right, I hope you enjoyed it, and I'll talk to you guys next time.